All right.
You.
Shit, I better see if this is live. Hi, everybody. So this is why I'm a terrible producer, because I can't really check if this is working because I can't do that many things at once. And yeah, it's a lot better when I have somebody to help me with this stuff. But anyways, welcome, everybody. I hope that I'm live on other platforms. I'm Dr. Angela Rasmussen. I'm a virologist, the co-editor-in-chief of the journal Vaccine. I live up in Canada, and I am representing public health and science on the Save America Movement Steering Committee. And today we are here, and I'm about to introduce you to my wonderful co-host in one moment. But we are here to cover the VRBPAC, those in the know call it the VRBPAC, the vaccines and related biologic products. Oh, I got it wrong. The vaccines and related, biologic products advisory committee, the VRBPAC. So what the VRBPAC does is it is a panel of independent experts from academia, from industry, from other parts of the government sometimes, or from people who used to work in the government, who are very, very qualified to evaluate different aspects of vaccination. And what they do is essentially similar to what the Advisory Committee on Immunization Practices, the ACIP, does for CDC, except instead of making recommendations that apply to the general public for what vaccines they should get, the VRBPAC makes recommendations to the FDA for which vaccines they should approve. And so every year in February, at the end of February, historically, the CDC would send its best flu people to the World Health Organization, where they would get together with all the other member states, compare influenza data, surveillance data from the prior flu season, and then they would select the different strains. Now things are a little bit different, and we're going to talk about that. But this process isn't different. So we decided that we would cover it because, unlike most of the other advisory committees in the federal government, which are total and complete shit shows, the VRBPAC is actually still staffed with a bunch of my wonderful colleagues who are actually quite capable and have quite a lot of expertise to make this decision, which is really consequential because it determines whether or not the FDA is going to approve flu shots for us next season. So let's get into this. Without further ado, the VRBPAC is on its lunch break right now, but we're still working. We're going to tell you all about the morning session and what to expect in the afternoon. This is my awesome co-host, Dr. Saskia Popescu. She is an epidemiologist, an infection prevention control expert, previously worked in one of the largest hospital systems in Arizona during the pandemic. She's also worked in the entertainment industry, providing guidance for movie stars and things like that. And now she is one of probably the most respected experts in the field of biodefense, biosecurity, and global health security. She runs a global health security network. And she is a good friend and colleague. And according to at least one anti-vaxxer, she's also my husband. So without further ado, welcome, Dr. Saskia Popescu, and thank you so much for filling in for my other wonderful co-host, Jessica Malaty Rivera, who will be joining us later.
Absolutely. Thanks so much for having me. And hopefully I can be entertaining, as I always forget that was the rumor, that I was your husband, but it's lovely to be on the show with you.
Well, I mean, we don't really discourage that rumor. Saskia and I have traveled quite a bit together to many of the same conferences. And because we actually like each other, we have shared rooms sometimes. We've also gone to the spa together. And on one occasion, at least, they did book us a couples treatment at the spa because they appeared to be confused about the actual nature of our relationship. But that's okay because, we share everything. And today, we're going to share our thoughts about the trivalent flu vaccine formulation. So I know, Saskia, there's so much stuff going on in the world, and not everybody pays as close attention to this stuff as I do. One of the things that I have become just very involved in is influenza vaccines. And one of the things that's actually really surprising in my career is that, I work on emerging viruses, and I never actually thought that I'd have to spend that much time thinking about things like seasonal influenza viruses, because we've done a pretty good job at controlling them. And the focus has largely been, making new vaccines. And now what's changed is that there's not a whole lot of money for research left. Essentially, the entire field is facing an existential threat. Regular influenza vaccines, which have been used since, for 80 years, have been relegated to this shared clinical decision-making status by ACIP and by Secretary Robert F. Kennedy Jr. of the Health and Human Services Department, the health secretary. He has just decided to make flu vaccines shared clinical decision-making instead of recommending them for everybody, which means that essentially you have to talk to your doctor before you should get a flu shot, which is ridiculous because we've got, again, 80 years worth of data. Mm-hmm. But what's really interesting about this, and why I never really thought about it before, is that we've been dealing with this for the last 80 years, right? Like influenza viruses, there are a lot of them. They change. As soon as everybody starts getting immunity to one of them, the virus itself will start changing. Flu virus has a lot of mechanisms for changing very quickly. And oh boy, check this out. I've got slides that move around. And so we can look at what influenza virus looks like. This is influenza virus. Influenza virus has a genome that's in 8 pieces, and these pieces can mix up with each other. It's called reassortment. When this virus reassorts with other influenza viruses, it can make very, very rapid changes in terms of how your immune system recognizes it. So what your immune system does is it recognizes these proteins called hemagglutinin and neuraminidase that are on the surface of the virus particle, which is called a virion. As these things change, and they change because there's mutation, as well as swapping these different genome segments back and forth, you end up with a protein on the surface of the virus that can look very different to your immune system. So every year, the GISRS, I like to pronounce that one with a hard G. See why for yourself. But the Global Influenza Surveillance and Reporting System is operated by the WHO. And back in the before times, what would happen is every country who is participating in this, which is most countries, submits all of their influenza data, including their sequence data, which shows what these proteins actually look like, to the GISRIS. And everybody gets together in February, reviews this, and then they try to predict essentially which flu strains are going to be circulating. Then they go to the WHO collaborating centers, which are a group of labs all around the world that collaborate, as the name implies, with the WHO. And they have these libraries of what are called CVVs, or candidate vaccine viruses, that they maintain. And then when this data gets recognized, they look at these CVVs, and what they try to do is they try to have one that is immunologically representative of those types of viruses. So hopefully they've got a CVV in there that's a very similar immunological match to the viruses that are circulating and that they think are going to dominate in this flu season. And so that's what this meeting is typically for. It's normally kind of a rubber stamp meeting, although they do a lot of evidence review. Basically what the VRBPAC does is they do the same thing that the WHO group did, except they just do it for the US. So they take all of that data, they review the safety and effectiveness data, they review the surveillance data, they review where the virus is in the world, they review different epidemiological patterns, which Saskia, I hope you might be able to tell us a little bit more about. Because one of the things about flu is that as it travels around, because it can reassort, it can make different viruses with different sort of immunological properties in different parts of the world. And that can have really different impacts on the healthcare system, particularly when the vaccine isn't matched well. And I know, Saskia, that you know a lot more about that aspect of it than I do. So that's why this meeting is really, really important. Last year, this meeting was canceled and not rescheduled. And it was decided that they would just go with the WHO recommendations, but it was decided in a closed door meeting that, not a lot of people saw or could participate in or had access to. So that was really concerning, because it's like, if they're going to do that this time, okay, great. They picked the WHO recommendations, but what are they going to do next year? So next year, I don't really know how this happened. That's this year, the meeting that's happening today. they actually didn't get rid of the experts on the VRBPAC. And so now they are going to go through the standard VRBPAC evidence-based review procedure, which is great news for this year's flu shots. It's not great news, though, for the years to come, because I unfortunately have a very bad suspicion that if they do just make a routine recommendation that's based on evidence, and so far the presentations have been really, really data-driven and really great in that regard, I think that they might just be like, well, this isn't doing enough to keep flu shots out of people's arms, so no more VRBPAC. Or they're going to do something like they did with ACIP, where they fire everybody and they replace them with a bunch of anti-vaxxers who are the least qualified people imaginable. We've had a lot of turmoil at the FDA. My friend Vinay Prasad, who I've been beefing with, and I know you have too, I think since 2020, one of our old colleagues from.
Yeah, he's one of my favorite people who just blocks people who don't even know who he is. I actually forgot who he was for a long time. And then when I looked him up, he had blocked me. So I was like, I've never even interacted with you, but some people just choose anger and violence.
Yeah. And I mean, that's a good way of putting it, because it seems that he was still so angry about this, not just from you, but from literally every single one of our colleagues who disagreed with him, criticized him, or said that he was wrong. That's been everybody's experience pretty much. But unfortunately, he ended up as the director of the Center for Biologics Evaluations and Research, which is otherwise known as the top regulatory role in the entire U.S. government for vaccines and other biologic therapeutics. He's the nation's top drug regulator. And basically what he started doing was overriding expert committees, including internal FDA committees, as well as committees like VRBPAC, and basically just more or less having a hard-on for randomized controlled trials, and deciding unilaterally that if the trial design hasn't, met his high standards, honed, I guess, from years of podcasting about them, then he's not even going to review the drug. And he did this to Moderna and their seasonal flu vaccine, which actually showed in New England Journal of Medicine report on their clinical trial that it was improved compared to conventional flu vaccines. That technology is much easier to manufacture quickly. So approving it by the FDA would provide actually a brand new option for seasonal flu vaccines that might actually work better. And for some people, I think, it would really work better. But for flu season in general, this season we had this H3N2 variant that emerged called subclade K. And it wasn't as good of a match for the vaccine as the H3N2 strains that had been circulating when the strains were chosen. So we couldn't have predicted that. And that happens. Evolution is messy and biology is messy, and it is not easy to predict. And that does happen from year to year. But if we had had mRNA vaccine technology, it is actually possible that when H3N2 emerged as early as it did, and it emerged in the Northern Hemisphere very early in August, that is the very, very beginning of flu season in the Northern Hemisphere. And that's very, very unusual. And that was a big topic of discussion this morning at the VRBPAC, which I watched so that you all didn't have to. I think that it's one of the things to keep in mind, and this is really where I value your expertise so much, Saskia, is that a thing, and this was a big topic of discussion. I did a lot of media interviews about this over this past winter, is that people started to think that H3N2, like the H3N2 component of the vaccine, was just completely worthless because it wasn't as good of a match. But the fact is that vaccine, and I think we're about to see more data this afternoon coming up at VRBPAC will show this, that even though it wasn't as good of a match in terms of assays that you can do with a lab, it actually still significantly reduced the disease burden, according to these early estimates. And the next session at the VRBPAC, which I'm going to be watching and doing commentary along with, possibly talking to myself if Jessica doesn't show up soon, although she said that she is going to pop in as soon as she is available. But that's what they're going to be talking about. They're going to have the Department of now War talking about their surveillance data. So in the US,
and they actually have very good
DOW
surveillance data. I just want to throw that out there. So like, I think it's interesting because, Angie, I think you had that slide up that WHO just released their influenza vaccine recommendations. So for this season, for this coming flu season. So I just want to note
wanted strain selection.
Exactly. So I am fortunate where I get to kind of I split my time between Arizona and Australia, which means I actually get 2 flu seasons. Lucky me. And Australia had a pretty unusual, very similar to the US, where it was a weird flu season. But the reason I say this is because that's where the first surveillance picked up this subclade K. But it was a really unusual flu season because while the peak wasn't as high as it was last year, it kind of went up and then it just stayed elevated for a much longer period of time. And then there was another very unseasonal bump up again. And so I think what worries me, and the reason why I say DOW has such good surveillance, is because through their global emerging infectious surveillance network, they actually are some of the best biosurveillance resources we have in the United States. And I think it's important because increasingly, as we have pulled out of the WHO, which we drastically rely on for surveillance of infectious disease, all of this information is critical for our response efforts. We're going to rely on systems like the CDC and systems, but networks really that are intrinsically built into organizations and agencies like CDC, which was really impacted with all the budgetary cuts, but now very much DOW. And I will say some of the most wonderful scientists are a part of their biosurveillance network, and they pull surveillance from all over the world through bases. They're increasingly doing a lot of really amazing environmental wastewater surveillance. But the problem is, though, that's still limited, right? So my concern, and I'm sure, Angie, you appreciate this because you help make vaccines and we need information to inform those decisions. So my concern is, as we're having organizations and meetings around what do we do next, that we're not making as informed decisions because we're relying on other people providing us what little information they're now doing because we pulled back so many resources. And those networks, that strength of a surveillance system is increasingly dwindling. And you've all heard that saying, disease knows no borders, which is true, but some borders are more porous than others. Some countries are more resilient, more ready, and have systems that are a lot stronger. And I think we saw with COVID, we can build a really robust system, but if you don't work and collaborate with others, it's only going to get you so far. So thanks so much for bringing that up, Angie, because I think that's such an amazing surveillance network. But as you can see here, it's not perfect, right? We still really rely on global partnerships and entities like WHO.
So Saskia, this was perfect timing for you to talk about that, because this is actually not my slides. These are Bill Gruner's slides from the Department of War. And as you were just talking about the value of these international collaborations, I couldn't hear what he was saying, and I'm about to turn the sound on so that everybody else can hear.
Yeah, please do.
But I think that it was just perfect, beautiful timing that you started talking about that right at that moment, because I'm almost positive that that's exactly what he was talking about. So without further ado, I'm going to turn this on. And so this is kind of an experiment, just to kind of get used to this format. We'd love to hear from people watching what they think of this. We kind of did this, me and Jessica Malaty Rivera did this for Casey Means confirmation hearing, where we were watching the hearing. And in that case, we were fact checking things in real time because, oh boy, did she say a lot of things that weren't true. I do not expect Dr. Gruner to follow suit with that. He is one of the colleagues that you just mentioned, Saskia, who is part of this really accomplished biosurveillance network. So I am going to turn on his sound if I can figure out how to do that. And
Angie, I can hear it, just so you know.
You can hear it? Okay, great.
Yeah. And we just lost Angie. Oh no. So, I mean, I hopefully she will come back in. I'm just going to ping her. And But I think the big thing that they're going to be emphasizing is pulling all of that biosurveillance data, that is including flu data. But just so everyone's aware, this isn't just flu that DOW does through GEIS. It's also emerging infectious diseases like Ebola and Marburg, everything from Mpox, but also to seasonal respiratory viruses. So it's a really important resource that allows us to keep tabs on kind of what's going around everywhere, but it is intrinsically limited to where we have those resources. So most of those are where we have a military presence and base. The goal is to increase those surveillance networks, but of course they do rely on lab capacity.
And adults with
Angie.
Just due to low numbers of those groups there that we can perform analysis.
Hi there. Sorry about that.
You're good. I was explaining how some of these surveillance networks
In all,
work.
we had 312 cases and 692 controls with 31%
Sir, I am.
and 69%
I
vaccination rates in these
was,
groups,
going
to be considered vaccinated.
pop
an individual
over
needs to
and
present
try
with
to grab
symptoms
another
14
slide
days or
and
more
figure out
after
like my
receiving the
sound
2025-26
situation, and I
Northern
hit the wrong
Hemisphere
button
influenza
and I
vaccine.
closed my browser.
In the figures on the right, you can see the breakdown
I'm
of the influenza
once
subtypes included in
again,
this study from
I
the
have
United States
demonstrated that I
and
am
from the
probably the
OCONUS
least
or outside the United States
computationally
locations in the
competent
top right,
systems
and
biologist in
total
the
on the bottom.
universe.
Next
In
slide, please.
fairness, I haven't been feeling very well lately. So
It's a quick overview of the study
not in great shape, not with flu, but
illustrating the slight majority were
forgive
female.
me.
Most tended to be
This will
under
be
the age of
something to work
18,
out
and
for
most of our samples
the
were collected from
ACIP meeting,
December through
which is
February,
next
with
week.
the highest number
And
collected in
I
January.
should just add right now
slide, please.
that I've already informed Saskia, much to her misfortune, I'm
So
going to
here
be
are the
pressuring
percent
her to
VE
join us
calculations and their
at the
confidence
ACIP.
intervals
But
from the study,
Jessica
with
Malaty
statistically
Rivera and I will be
significant
once
results in
again
red.
covering the
Adjusted
ACIP.
VE
And
ranged from 18
unlike
to
this meeting,
75% across the
where we're
various
seeing a lot of
categories.
really
VE was moderate
fascinating
to high
and
for
I
AH3N2
suspect
and
extremely
significant in all
accurate
the dependent groups for H3,
vaccine effectiveness
with
data right
75%
now.
VE for
These are the
adults being the
mid-season flu
VE
estimates
was
where
also
every
high and
year
significant
the
for influenza
CDC
B with
with
all
their
dependents of
respective data
63%,
and the
and
military with
higher
their
generally for adults in
respective
all the categories
data
were
from
available.
service people and their families
Results were not
primarily
significant for any of
will
the
calculate
AH1N1
at various
PD-L1 or
points
for
during the
children,
flu
the
season
overall
will
influenza
calculate the
A, any
effectiveness of the
subtype,
vaccine. They
or
also
the
calculate
influenza B
things like
category.
adverse event rates and
Next
stuff
slide,
like
please.
that. This is,
So
a
this
graph
slide
that
just shows a
shows
forest
pretty
plot of the
much
same
how this
adjusted VE
looks from
calculations for
season to
easier
season.
visualization and
And
comparative
where I was
results.
kind of going
The
before we
confidence
went
intervals
off on the
between the different
Department of
groups.
War, Saskia,
Next
was
slide, please.
that even when a flu vaccine is not well matched,
So we'll now
and you see
transition
some
to
of this
VE
stuff, and I
estimates
think
against
that's exactly what
medically
we're
attended
about to see
ambulatory
here.
influenza infections among
We're
active
about to
component US
see
service
this
members.
data that he's
Next
about to
slide.
show us is going to be from service
study was
members
also a
who
test
got
negative, safe,
sick,
controlled
went
design for
into
outpatients.
various, military
This was done
hospital
using
or
active
military
component
clinic,
personnel from across all military
got a
services,
test
including
for
recruits,
flu or
and
were
included those
diagnosed with
stationed in the
flu.
continental
And
United
then that
States,
gets
as well as those
reported, and
stationed
then
in
they
foreign
look at all of
locations
these different
from
features
December
and they
1st through
figure
February
out
11th.
how many of them were
As in the
vaccinated
beneficiary
and how
studies,
many
influenza
severe
cases
cases
were
the
confirmed by
vaccine
PCR
prevented, how
culture,
many cases the
but in
vaccine
this
would have
study,
prevented
rapid test
overall.
positives were used to
When you
define
design
cases
this
as well.
trial
While
correctly,
controls did not include
it's pretty
negative
easy to
rapid
find
tests.
that. And what's nice about the
Models in
military,
the
as
study
you were
were
saying,
adjusted for
Saskia,
age,
is
sex,
that not
prior
only do they
vaccinations,
track
and
their
prior
service members'
in
health
previous 5 years,
pretty
and month
closely,
of diagnosis.
they also require
Next slide.
service members to be vaccinated. So they have
So
a
here you can see the
population of
age
people where they
distribution of
have
cases
really,
and
really
controls in this
good
study.
historical
The large
vaccine
numbers
data on
were
them,
from the 18
and they
to
have really,
24
really
year
good
age
vaccine
group, which is
uptake because
interesting
it's
because
required for
in
service
previous
in the
active
U.S.
component VE
military.
studies, the largest
And
age
so
typically
you are
is in
able
the
to
30 to
actually
39-year-old
do
range.
stuff like this, which
Next slide.
I see I'm pointing at my other screen here, but
Most of the
you're
vaccine
able to
types
do stuff
included in
like
the
this.
study
We were
were
able to
IIV.
calculate
Most of the
because you
subjects
know
were
exactly how
vaccinated
many
previously within
cases
5 years,
got all
and
these
most of the
different
cases were
subtypes of
influenza
flu,
A.
or just had flu A
Over
or flu
700
B.
cases of
There's two flavors of
AH3N2
flu
and
essentially.
influenza B were used.
How
However,
many of
only
them had a
51
prior vaccine? How
AH1N1
many of them had
PD-L1
recently been
cases were
vaccinated?
available
That
for the
sort of
study.
thing. And they can
Next slide.
figure out how well the vaccine is working from year to year.
Here we show
Now,
the
usually
VE
the
estimates
vaccine,
against both influenza
you
A
hear
and B, as
anti-vaxxers
well
all
as
the
A
time, and you even
subtypes
hear some of our
H1N1,
colleagues
PD-L1 and
who tend to
H3N2.
focus on the years where the
Once again,
vaccine really
statistically
doesn't
significant
work very
estimates are
well.
in red,
And there
showing
are some
moderate,
years like that,
adjusted VE for
where,
H3N2,
it doesn't prevent
and
that many
moderate to low
cases, which
protection
is
for
really
the
the
all
main
influenza
metric
and
that they
influenza
use
A
to figure
all
out how well the
subtype
vaccine
groups.
works.
There's
But I
also
think
a
one of
forest
the
plot here
things
for
that
easier
really strikes
visualization.
me, and this goes
Next slide,
to
please.
the military in particular, because the
So now
history
I'll
of
transition
flu
to our genetic
vaccines
and
is
antigenic
actually really
analysis.
intrinsically linked to the military. This is something that I think about all the time, is how many instances throughout history that American freedom, essentially, and democracy was upheld because of vaccination or immunization. Like, so we won the Revolutionary War, arguably, because General George Washington mandated variolation, or at the time it's infecting people with smallpox on purpose so that they don't get really bad smallpox. They didn't know that much about immunization, and they hadn't discovered vaccinia virus yet. But in any case, George Washington immunized the Continental Army against smallpox and won several decisive battles. It's entirely possible, I like to think, that immunization won the Revolutionary War. In World War I, the death toll from the 1918 flu pandemic was much higher than World War I, which is generally considered the Great War to be like the worst of the world wars. And you know it's bad when it's worse than the one that had the Holocaust in it. But the U.S. lost almost 50,000 troops as a result of the 1918 flu pandemic. And as a result, by the time World War II came around and they had started to develop inactivated vaccine technologies, they said, hey, I think we really need to make this a military priority, because we can't have like all of our troops in Europe dying of flu again. And so they tapped a guy named Jonas Salk to grow up a bunch of fluid eggs, inactivate it. They did a clinical trial in military service members who were associated with U.S. colleges and universities. And they showed that inactivated flu vaccines work. And those were our first flu vaccines. So you move ahead to now, and you are mandating, vaccinating our military service people. And you can say better than anybody, Saskia, what it's like to have a bad flu season in terms of the hospitals and in terms of availability of healthcare. And as a national security issue, I mean, I don't think this gets talked about enough. I don't think people understand how destructive and harmful it is to national security to have our hospitals full of people and all of our healthcare resources being utilized when it's civilian healthcare resources. But I also don't think in this age of we're starting a new war every week, how detrimental it is to our national security to have those hospitals full of our soldiers.
I think one of the big things is that, force readiness comes into play, right? So this is keeping our soldiers safe. But I think we often kind of create a dichotomy where it's like the civilians in the military. But keep in mind, our military, these are still people in boots. They're still humans in boots that go home to their families. And if they get sick, it's not just a national security concern because it impacts force readiness and resilience, but they come home and they can get their families sick. And that can overwhelm public health, healthcare, whether that's the VA or just our hospitals everywhere. And I do want to note, we saw with COVID-19, there were several outbreaks aboard aircraft carriers and naval ships. I think it was the USS Roosevelt that had a really significant outbreak of COVID-19. And so we saw in real time how a respiratory virus can just roll through a single naval ship. And I think these are environments where respiratory pathogens can spread so effectively and really take down everyone. And if we've all been sick, even if it's just a kind of a crappy cold, you're not effective in your work. You're stuck at home, and that can be really, really debilitating. So these are environments, military barracks, ships, Humvees everywhere, where it's not surprising that respiratory viruses can spread easily. So it does become, I think people kind of laugh, oh, it's just flu. But if you lose an entire ship or convoy because of a respiratory virus like influenza, that can be very impacting to that mission. So I'm sorry, I'm gonna have to love you and leave you, Angie, but I think that's a really important note, is that at the end of the day, this isn't just, seasonal flu. It really does become a public health issue and a national security issue, which I do want to remind people, public health is a national security issue. That really is.
It really is, Saskia, and that's one reason why I wanted you to come on today. I'm so glad that you could come on, even just very briefly. Really appreciate it, because while I can sit here and I'm about to tell everybody what antigenic cartography is, it's actually very fascinating. But I think your perspective on this is so
So this is
important
just the
because
titer
what I
table of
just
the hen
keep
data that
thinking
was
of
used to generate the antigenic
is
cartography
how
on the previous
destructive
slide.
all of
reference
this is
antigens
going to
here
be.
are
So all
across
of the
the top,
preventable
and then the
diseases that are
vaccine
coming back, you know,
or
measles,
the
flu,
vaccine
all of
CVV,
these
and then the test
diseases are
reference
respiratory
viruses are
diseases.
down the left side.
RSV, which at the
So while both the
ACIP meeting,
2025-26
it's expected that
vaccine
they're
strains
in some ways
orange
going to go
and the
after some of the
2026-27
RSV immunization
WHO
And
options.
the
that are
numbers are
available.
climbing right now
green
in the
shows
US.
good
RSV
protection
numbers are starting to
against
climb.
D3.1
So
and
They're
D3.11
climbing, and you know what?
strains
We're
with
going right
generally
into
moderate
flu B
protection against
season.
the other subclades.
So a
You
lot
see
of
that
people don't know this, that
Missouri
there's also
11 in
flu
the
B,
green has
which
a slightly
starts
higher
off
number
usually
of
in the
strains
spring.
that fall within an full
There's a
threshold.
ton of respiratory
Next
diseases,
slide,
and
please.
you know, I spent a lot of time thinking about what
So for
happens
the
if
H3N2
H5N1
HA
acquires
segment,
the
we saw
ability
an
to transmit
overwhelming
from person
majority
to
of
person. We have
subclade
a bird
K,
flu
which I think will
pandemic,
come
and
as
that's
no
really
surprise to
bad.
anyone at this point.
But how
That's
much
represented in the
worse would
purple
it
on the
be
tree.
if we
So
had a
similar
bird
to
flu
other global
pandemic,
trends, we
even one
continue to
that
see
didn't
very
have a
low
very
level
high
circulation
mortality rate,
of the
but that
other
put enough
subclades, the
people in the
J2,
hospital
JT2, JT3,
that,
and
you know,
JT4.
the hospitals are already
And then
overwhelmed
similar to
because,
the
you know,
H1N1
there's
tree,
thousands of measles
the
cases now,
H3N2
and we
2025-26
only have so
vaccine
many of these
strain, the A district of
resources. And I
Columbia
just
you'd
don't
be
27
think
in
that those,
surprised
2023,
how easily a
the 10 in
hospital
red,
can be
and then the
overwhelmed. And I think
2026-27
that's the thing
WHO
people don't
Northern
realize
Hemisphere
is
strain
we every
recommendation,
single year in a
the
hospital, we kind
ADR1
of brace for
1415
impact. We start
2025,
really in
and
October.
ADR1
And whether that
14
is, you know, in
2025
pediatrics, we had
are
seasonal
shown in
visitor
orange.
restrictions because it was just that we
And again,
could
the
not
bars on the
handle
right
people bringing
show
in
the high and low
siblings,
reactors to
especially because, you know,
DC27 and A
love
Wisconsin
kids, but a lot of
114
times they bring in a
2025,
bug
which
to the
is an
hospital and get
114
people sick
15
there. So whether that's
2025
visitor
blood virus,
restrictions or just
it's
really knowing
suggesting
we're going to see an
fewer low
uptick
reactors
in
to the
respiratory virus illness
2026-27
and
WHO recommendation.
hospital census is going to peak. It's going to be
The
really
rest of the
hard.
circulating strains
We're going to have
here
healthcare
shared
workers sick,
loss of
and this is a
glycosylation,
time I
aside from
cannot say it enough that
C175A,
it strains our healthcare system so much, and
and a
that's
gain of
just regular. So
glycosylation aside from
if we don't
S146A.
get that kind of break in
We
between,
did have several
or
one-off
it gets
changes to
worse than we're
glycosylation
used
sites,
to,
but it
it's
is too hard to fit
especially
in
right
brain
now when we have
comparison
burnt out
variables.
healthcare workers, we're seeing hospitals close. We
We did
really
not
can't sustain that.
observe any
It's not
consensus
something that
presentations.
I worry. I genuinely
of
worry
any
for the US
antiviral
healthcare and public
resistance.
health
Although again,
infrastructure.
we have
So
minor variant
with that
analysis
cheery
for
message, I'm
phenotypic
going to leave
testing
everyone in
performed.
the very amazing, capable
We did have
hands.
23 viruses that
Hopefully
shared the
Jessica arrives soon so
S59V,
that we you alone too much, but
S144N,
thank you for having me. I appreciate
M158B,
it.
I160K, and
thank you so much for coming
T328A
in
were
and
mentioned by Dr.
providing me
Conner
with somebody to chat with.
Next
As
slide,
you
please.
know, I
Always.
can chat with myself all day long.
And so, again,
So,
this
but
slide
it's
just
Thank
a
shows
you.
lot
the
more
D2
interesting to
clade
chat with you,
proportions throughout the
Saskia.
season,
So thank
both
you
as
so
the
much
total
for
number
sharing
on
your
the left and
expertise.
then as the proportion
We'll
on the
see you
right.
next week
And you
for
can
the
see the
ACIP
overall
Definitely.
meeting.
dominance of the subclade
All right.
Have a good day, everyone.
However, we did
Have
have
a
other
good one.
subclades such as JT4 that have still remained.
All right, guys.
even recently.
We'll get right
Next
back
slide,
into
please.
this. So, you know, again,
And
let
it's
me
the
know what you
antigenic
think
map for
about all of
H3N2,
this. I
as
mean,
with the
if
H1N1
you know, this is
cartography, the
something
small
that you find it just
anti-sera.
intolerably
Circles
boring.
represent circulating
We can kind of
viruses,
come up
and the
with
large
another
colored
format,
circles
but
represent the
what I
reference
liked
viruses.
about this format,
Subclade K
and I
viruses are
especially
colored
like
red
it when I
here,
have
while the
somebody like
J2
Jess or
viruses
Saskia here
represented by
to
the
bounce ideas off
2025-26
of,
vaccine
because
strain selections,
just my
A
perspective,
Croatian 101
believe it or not,
36LV,
it even
and
gets
A
boring
District
to
of Columbia
me.
27 2023
But
in
you know,
blue.
I could like look at
Followed
this
by
stuff
other
all day. I
colorations for the
love
remaining
me
J1
some
and J2
influenza
subvariants.
virology, and I love some virology.
So here the large
So you're just
circle
stuck with me
represents
until
an
Jessica
eightfold
shows up,
difference
but
from the
hopefully
anti-sera to the
this
cell.
will be a little
A
bit
Wisconsin 114
educational for people,
2025.
and
As a
hopefully will be a
reminder,
little bit
is a
engaging
Darwin
too.
1415
One of the
collector
biggest
strain.
problems, I
And you can
think,
see
is
that
that
circle encompasses most
to
of the
me, this
subclade K
graph
viruses and
that
many of the other
that is
subclade
being
viruses
presented
pretty
right
well.
now makes a lot of sense.
Okay, next
What
slide,
this
please.
graph shows is where the subclade K
In this titer table for
viruses are falling
H3N2, you can see that
evolutionarily. This
Michigan
table
105
is
2025
showing
and the
essentially
cell A
how many
Wisconsin
antibodies
114 2025
these
in
different
green,
subclade K
which once
viruses
again
will
are
induce.
Darwin 1454
Most
and
people
Darwin
will look at
1415
this
like
and be
viruses,
like,
respectively.
boring table. Before that, there were a bunch of
Generally have higher
dots.
titers than the 2025
What the hell does that
vaccine
mean?
strain selections in orange.
You know,
And they have
you don't
fairly broad
necessarily
coverage
need
across
to
most data
know
subclade
every
viruses, especially compared
dirty,
to
like
anti-sera
nitty-gritty
raised against
technical
the
detail
JT1
of
through
what
4.
this all means. But you
Next slide,
should
please.
know that this is how we actually
So
get
lastly, we
access
have
to
the
any
analysis
kind
for
of
B
flu
Victoria,
vaccination.
starting with the phylogenetic
These
tree here.
vaccines,
The majority of the
you know,
sequences characterized
they don't
belong to the
just sort of
subclade
get picked out of a
C3.1,
hat every
while we
year.
also did
We
observe moderate
really have
circulation of
people
C3
all
and
over the
C5.6
world and
and
all
low-level
over the
circulation of
country
C5.1,
who
C5.61, and
have
C5.7.
dedicate their
Most of our B
entire,
Victoria viruses were
their
collected in the
entire
United
year
States, therefore this
basically
higher
to
proportion
following
of
all these different
C3.1
viruses and
highlights
doing
Dr.
things
Condo's
like this, which is
observation
making a phylogenetic tree that shows essentially the direction that all of the circulating viruses in the human population are evolving in. And this one is for influenza B, the Victoria lineage in particular. So influenza B is really important. We don't spend a lot of time talking about it. It's not H1N1 or H3N2. It's an entirely different type of influenza virus, although it is still an influenza virus. Influenza B viruses aren't as common, but they can cause really big epidemics. And so for that reason, the third component in the influenza vaccine is an influenza B component. Now, interestingly, this goes to show you that things like lockdowns and public health measures actually do work, because during the COVID-19 pandemic in 2020, influenza B virus Yamagata lineage was rendered extinct. Nobody got it because so many people were staying home that there wasn't a critical enough mass of potential people that could be infected with Yamagata that it was able to actually spread through the human population. Influenza B viruses are a little bit different in terms of their ability to infect other animals and cross species. So the fact is that people weren't getting Yamagata, they weren't transmitting it to each other during the pandemic, and Yamagata went extinct. It has not been seen since 2020. And then it was seen very, very little. So we actually got rid of one of these influenza viruses, or a particular lineage of the influenza B viruses, just through imperfect vaccines that don't work pretty well, or don't work perfectly, as well as staying home and not interacting with people as much. So this is something that, you know, to me is one of the most incredible stories that a lot of people just don't know about, because these flu vaccines, which don't work perfectly and don't prevent infection, combined with public health measures, non-pharmaceutical measures that do not specifically provide protection like a vaccine would, combined, those can still actually eliminate virus transmission. So that's why it's still important that even though the vaccines aren't perfect, that people continue to get vaccinated. And as I mentioned, the flu vaccines have three components. It's called a trivalent vaccine. So what he's talking about right now are the antibody titers. Again, this is what's called a hemagglutinin inhibition assay. It basically shows how many antibodies get induced by these different viruses and how well they neutralize the viruses as opposed to just binding them. So this is the kind of data that the Department of War, Department of Defense, and the CDC will be collecting year-round. This is also the type of data that is provided to the WHO GISRES, the Global Influenza Surveillance System, as well as the data that is, as we see now at the VRBPAC meeting, being used to make a decision about whether or not the VRBPAC is going to recommend the FDA, or sorry, the VRBPAC, the WHO strains that were selected. See, this is why it's better for me to have somebody to talk to, because then I don't just start like using words that don't make any sense. In any case, you know, this is the normal process that we would go through every year, and that we are going through this year, fortunately. This process is really important, even though it doesn't change that much from year to year for flu vaccines, because we've been using them for the past 7 decades or so. Now, the flu vaccines are pretty basic. Most of them are inactivated. They are grown either in a cell, in cell culture, or they are grown in chicken eggs. There is one type of vaccine that is a recombinant vaccine. So it's protein, essentially. It's that hemagglutinin protein that I mentioned, the H and H1N1 or H3N2. That is just expressed and injected. And so that's another vaccine. It works about as well as the inactivated vaccines. Previously, there's also been a live attenuated vaccine. It's still available, called it's flu mist, essentially. It's intranasal, you inhale it. In years past, it hasn't always worked very well. It's recommended for people very specifically, depending on age and prior risk factors. I mean, it's recommended broadly, depending on which country you're talking about. But generally, the inactivated and recombinant vaccines are the most widely used.
So with that, I want to thank you all for the opportunity to be here.
This is why it's really important with these different vaccine platforms different viruses circulating around,
Thank you very
different
much
degrees
for
of protection
clear
against infection.
presentation.
And that's actually different than
Questions,
protection from
please
disease.
raise your
So
hands.
there's a lot of things to consider, and that's what they normally do.
Dr.
And
Gans.
yeah, I'm going to be quiet for one moment because I can't actually hear the sound. And then I'm going to add this back. So stand by. Technical difficulties are not my forte at solving them unless it has something to do with the containment lab, which fortunately this does not.
In the give report. But a couple of things that are occurring here is you can have cells in your immune system that can stir up sort of other antibodies to other subclades. And so we have in the DoD, highly vaccinated population that gets vaccinated every year sequentially. So I think there's a lot of humans aren't ferrets. So the ferrets are naive. So you're looking at purely naive differences between the different clades antigenically there. Whereas once again, with the humans, you can have all sorts of other antibodies produced with that. So.
Thank you. Not seeing any other hands raised. Oh, Dr. El Sali, last minute.
Yeah, just a comment actually to Dr. Gruner and Dr. Gans in that also the UK observed, I'm not going to say high VE this season against the.
I don't think it was that high.
No, it was comparable. I was going to say comparable. I was expecting it to be low given the case circulation, but it was comparable. So and that's why I was asking if, I don't know, the end didn't drift as much or what happened. But
I don't know if we've seen later that was a really preliminary estimate. And I don't know what's happened since.
The one in the UK?
In the UK.
Yeah, I don't think there was an update. It was prelim like the one we saw here for the United States, and the VE was comparable despite the circulation of the K. That's what I was gonna say.
Yeah, and similar to what Dr. Grosskopf has mentioned, this is a mid-season VE analysis, so still a lot more data to be collected this season, so things can certainly change by the end of the year.
Dr. Gans, do you have your hand up again?
I do. Thank you. I was going to let other people have a chance, but no one's coming up. Can you describe a little bit then the individuals who actually did get disease in your population?
H1N1 type. For Southern Hemisphere seasonal influenza vaccine, the recommendations were for egg-based vaccines was a Missouri 11-2025 H1N1 PDM09 like virus. And that for the cell culture based was cell culture, recombinant, as well as nucleic acid-based vaccine. The same recommendation was Missouri 11-2025. Data collected and reviewed by the GISL system was
Okay, everybody, I am so sorry. I ended up having major technical difficulties in terms of my being able to not be able to figure out how to unmute you specifically. I really truly felt like one of the people at the ACIP who is constantly fighting with Robert Malone to have control of the Zoom muting. In any case, let's get right back to the VRBPAC. This is Beverly Taylor from CSL Seqirus. They're one of the largest flu vaccine manufacturers in the US.
Transition to TIV. We'd like to recognise that it was actually the VRBPAC committee that really pushed for this transition, and the transition, as with all things, was quite complex for us to achieve. There were lots of changes.
So one thing that Dr. Taylor is talking about here is the transition from a quadrivalent vaccine that included the influenza B Yamagata component to the trivalent vaccine that we are now using. Because Yamagata was essentially disappeared after the COVID-19 pandemic due to the combination of public health measures and vaccination campaigns that occurred, we don't have to have a quadrivalent vaccine anymore, meaning we don't have to have a vaccine that has 4 different virus components in it. We need to have a vaccine that has only 3, because we aren't going to encounter influenza B Yamagata lineage anymore in the wild. The virus didn't have any hosts in 2020. It could not proceed. It could not continue to survive, essentially. It couldn't make more viruses. So it's gone. Vaccines work. What do you know?
Approved vaccines for ex-US countries did not transition to TIV as quickly, and so they continued to make a recommendation for quadrivalent for those countries. So we did, through this collaboration, successfully transition to TIV in the US for the Northern Hemisphere 24-25 influenza season. I'd just like to highlight that the US was the only country to transition in this time frame, and that was made possible through the agility demonstrated by the regulators and the regulatory system in the US, and the strong partnerships across all stakeholders, including manufacturers. I'd also like to highlight that there were no delays to the season's vaccine supply through this transition. Next slide, please. So for the Northern Hemisphere 2025-26 season, the WHO recommended a change from the previous Northern Hemisphere to an A Croatia for the H3N2 egg component of the vaccine, and for the cell or recombinant platform vaccines, a change of the H3 to the A District of Columbia.
So one thing I want to point out is that Dr. Taylor here is talking about how the WHO strain selection process resulted in a recommendation to switch what's called the CVV, the candidate vaccine virus. So these WHO collaborating centers have these collections of viruses that are optimized essentially to go straight into vaccine manufacturing. Beverly Taylor works for one of the largest vaccine manufacturers in the US. And so as a result, the manufacturers play a really, really important role in this part of the process, because they need to know which viruses they actually need to grow up huge numbers of. In the case of Seqirus, they are making largely cell-based and recombinant vaccines, or cell-based vaccines. So they have to grow up a lot of different cells to grow the viruses in. That's going to be different from a company that uses an egg-based process in which they have to acquire a bunch of fertilized chicken eggs. For both of these, they might need to use slightly different viruses, because as viruses grow in eggs versus in cell culture, and it's usually kidney cells from a dog that are used, MDCK cells are sort of the standard cell type, Mann-Darby canine kidney cells, or the standard cell type for growing influenza vaccine viruses, they can acquire other mutations as the viruses grow in either eggs or cells. They acquire mutations that adapt them further to those particular host cells. That can change the way that the vaccine actually influences your immune system. So it's really important when the WHO makes these recommendations that they're accounting for the manufacturing process. And it's really important that the manufacturers understand the evidence basis for making these different changes. So this part is, it seems like, oh, well, what's the difference between, Missouri and Singapore, or Missouri and Sydney, or Missouri and Darwin, kind of things. And these are all just different influenza viruses. So they're H's on the surface of the virus particle, which again is the major antigen or the target for the vaccine for the immune responses that the vaccine can elicit. These viruses all are named after different places, but they really represent different proteins, and the immune system will recognize differently. So what Dr. Taylor's going over right now are some of those changes that the manufacturers have to be aware of in terms of making this next season's flu vaccine supply.
Well, for the H1N1, there was no change recommended. There were CVVs and potency assay reagents already available. So we're in good shape for the H1N1.
Should add potency reagents refers
the strain was
potentially
updated
to adjuvants and to other
we did have CVVs,
things in the formulation that are
which were
part of making the vaccine
we had been able to
equivalent
do
potency from one person to the next.
on those viruses.
Also trying to make the vaccine as potent as possible because these are inactivated vaccines. They're not as immunogenic as a live attenuated vaccine or a virus that would actually replicate
CVVs.
So as a result, you want to make sure that they're as potent as possible.
But potency assay reagents will be prepared once the suitability of the CVVs are confirmed.
Right. And I'm sorry, in this case, they're talking
Next
about
slide,
potency
please.
assays to confirm that the vaccine is actually potent.
I just wanted to show this slide again. We have used this slide before, but really, I want to highlight a couple of things in us moving from TIV to QIV. We're still carrying out some production at risk prior to the strain change recommendation. We're doing less than we did before when it was QIV, but in order for us to supply the required number of doses, not only for the US, but also globally, we have to start some production at risk. So that is still the case. And then the activities which are highlighted with the double-sided red arrows are just indicating steps in our process, which still take the same length of time, whether it's trivalent or quadrivalent vaccine. So if you look at the top arrow, this is looking at, with the strain change, we still need the production and standardization and calibration of reagents for those new strains. And with 2 strain changes, it doesn't matter if that's trivalent or quadrivalent, that production time and calibration time remains the same. The other area that remains the same is everything from the point at which we've made each monovalent separately. So formulating the final vaccine, the filling and packaging, the regulatory approvals and the distribution all take the same time, whether we're supplying a trivalent or a quadrivalent vaccine. So I just wanted to highlight that because, it's not that we've got 25% less in the vaccine and therefore it takes us 25% less time to make it. It's not that simple. So I just wanted to highlight that point.
This was a really, really great slide. It goes to show that there are parts of the vaccine manufacturing process that you cannot speed up. We cannot change these overnight. And I really, really want to drive home the point that the timing of approving and then manufacturing influenza vaccines is absolutely critical to their effectiveness. If you don't start vaccinating people in September-ish, you don't have vaccines ready to go at that point. You don't have them ready to ship by like the end of August. Then you're not going to have seasonal flu vaccines that are going to be available to people to actually get in their arms. Vaccines themselves are completely useless. Vaccination is what makes an impact in public health. And so having the manufacturers talk about which parts of their process cannot be changed, having them talk about what their production risks are, what risks they're willing to assume, making sure that they have all the information they need to begin the manufacturing process in a timely enough manner to have those vaccines be available at the beginning of the influenza season. All of that is essential. So when people talk about manufacturers really being money-grubbing companies, I mean, making flu vaccines is not only extremely high stress year to year, because if you miss any of these deadlines, then forget it, you don't have any flu vaccines. They're not really very profitable, and they're not as popular as they should be. So this is really a commitment that these companies have made to public health, you know, for not very much profit, actually. It has been very challenging in the current environment where basically the entire system and the regulatory apparatus is dominated by anti-vaxxers, for them to have it be even feasible to make the vaccines. It's not that they're going to lose a lot of money. It's that their business cannot survive making vaccines if they are not able to do certain things. It's really, really important that the FDA make timely evidence-based decisions so that manufacturers are able to actually make these vaccines and get them out.
Year on year, we've seen a decrease in the number of vaccines distributed.
This is also really important, so I'm going to go away for a minute.
Supplying approximately 40 million doses less than we were in the 2021-2022 season. Next slide, please. So I wanted to talk a little bit about the access and benefit sharing systems, you know, recognizing that the US is not a signatory to the Convention of Biological Diversity, but these access and benefit sharing systems impact all.
So there's going to be a whole separate conversation about access and benefits sharing. Oh boy, this is like a very challenging topic. I do want to just reiterate, however, the importance of support, regulatory support for the manufacturers. I think that this really, really underlines how dependent the entire vaccine system is on manufacturers to be able to make vaccines at all. The US government, other governments are generally not in the business of making vaccines. So we really, really do need to hear the manufacturer's perspective because they are an essential part of our public health system.
But we actually use a lawyer through the International Industry Association, the IAPMA, to check what the current status of Nagoya legislation is in countries, particularly if we're seeing a CVV from that country for the first time. The supply of influenza viruses is impacted by this, and since 2018, we've now seen over 40 seasonal viruses being delayed, which reduces our choice of which viruses we use. And for the Southern Hemisphere 2026, there was a recommendation for an A. Missouri like virus. So the A. Switzerland virus was listed by the WHO as a light virus. And this, we actually didn't experience any delays for this approval because the Swiss system, number one, had been used before. Two, is very clear, and the instructions and the forms you have to fill out are very straightforward. And there's also no negotiation needed because the Swiss authorities do not want any benefits for the use of the viruses from their country. They just want to track who is using a genetic resource from their country. It's really difficult keeping up with all the legislation because there are national laws, and the status and the laws in each country is not kept up to date. And many of the delays are also experienced to a lack of resources in some countries. So it's something that we face every season. We've got a system for dealing with it, but it is a hurdle for us to get over. In late 2024, this new multilateral mechanism for the use of digital sequence information came in, and companies are expected to contribute either through this system or to comply with the national access and benefit sharing laws, because some of the Nagoya laws actually include DSI, and the contribution rates depend on the company size. With business sectors that heavily use DSI, which includes vaccine companies, a suggested rate of 0.1% of total revenue or 1% of total profits, regardless of whether DSI was used for every product. So this is an indicative rate at the moment, which means it's not finalized. But I just wanted to highlight this because these are some additional hurdles that we have to get over before we can, we know we have legal certainty to move forward with certain CVVs. Next slide, please. So on these access and benefit sharing systems, you know, the bottom line is that rapid and unhindered supply of influenza viruses and their sequence information for both seasonal and influenza viruses with pandemic potential is critical if we're going to be able to prepare the vaccines in time. And this global access and benefit sharing landscape is becoming extremely complex. So as well as the CBD systems that I've just mentioned, Nagoya Protocol and this DSI mechanism, we have the WHO pandemic agreement being negotiated, and we still have the WHO pandemic influenza preparedness framework, which impacts all manufacturers. And this does cause delay in virus sharing. I'll just give one really quick example. The PIP framework supposedly should enable us to access influenza viruses with pandemic potential without any hindrance. However, depending the route at which the virus is obtained, so if it hasn't come through the WHO GISRIS and it's come through one of the animal surveillance route, it actually falls under the Nagoya Protocol. And we have one case of a virus from Africa, an H5N1 virus, which was recommended as a CVV by WHO in September 2022, and we still do not have access to that virus. So this is a real issue that we are tackling. There's some acknowledgement of the need to prevent overlapping requirements between all these systems, which are referred to as instruments, but it's unclear how this will be achieved in practice. So what we don't want is delay after delay because we're fulfilling the obligations in each one of these instruments. We do continue to be very active and engaged in discussion.
Okay, I just wanted to butt in here and say that this is a hugely important point. As Dr. Taylor was just talking about, the issues surrounding benefit sharing are incredibly important, incredibly consequential, and also like very, very, very infinitely complicated. Basically, what she's saying is that the CVVs that are used to make every influenza vaccine, including emergency influenza vaccines like the H5N1 vaccine that Securus also manufactures, where there are 10 million doses in the US strategic national stockpile. There are also 500,000 doses that have been purchased of H5N1 vaccine for Canada. These vaccines were made essentially with CVVs that had been selected by WHO and recommended for some of these emerging H5N1 viruses that have come out. So this is really important also to have vaccines for emergent flu viruses that we might need a lot of vaccines for very quickly, because it takes about a year, as Dr. Taylor showed a few slides ago, to actually manufacture these vaccines at national scale, much less global scale. So it's really, really important for manufacturers to have access to a full library of CVVs, so there are as many different vaccines as possible available, or vaccine viruses as possible, that are available to the manufacturers, so they can get this process started as soon as possible, and we can actually have vaccines at national scale should a big virus emerge. So you also want to do that while making sure that the countries that are providing these viruses to the global system are given proper credit, and also that they have intellectual property rights for what they've contributed, and that they are being able to get equitable benefits, much like the wealthier countries like the US, Canada, and the European Union do. So it's a hugely important issue, and it's complicated by this whole complex ecosystem of different treaties and agreements and instruments and whatnot. The bottom line, though, as I think Dr. Taylor is talking about right now, and I'm about to shut up and unmute her, is that manufacturers play an absolutely critical role in us having vaccines. And we need to stop thinking about manufacturers as adversarial. The FDA in particular needs to stop treating manufacturers as adversaries and start treating them as collaborators.
The vaccine uptake and vaccine coverage rates have declined year on year. I've just highlighted the concerns of the Nagoya Protocol, the DSI mechanism, the pandemic agreement, and the lack of legal clarity and the risk of us seeing stacking obligations with these agreements, and the impact that that has on the supply of seasonal and pandemic influenza vaccine manufacturing, and how rapidly we're able to respond in an emergency. And the final point, just to reiterate again, that the global scientific collaboration and transparent sharing of samples and data is critical for public health decision making. Next slide, please. I think it's just, thank you so much for your attention and the opportunity to present the manufacturer's perspective. Thank you, Dr. Monto.
Thank you so much, and thank you for bringing up some important elements and complications that we don't generally think about or even discuss here on the VRBPAC, because really things like the Nagoya protocols have been lurking around for quite some time and have never really been fixed in an adequate way. Questions from the group, please.
I'll add the question and answer sessions this morning have been on fire if you're a scientist who likes to hear about flu vaccine data. Not so much maybe if you're a member of the public, because they show.
Thank you so much for that. I agree with Dr. Montoy. It's really helpful for us to understand all the perspectives on this. You talked a lot about timing, having enough time, using current data. Do you find the timing of this meeting in particular helpful to that timeline? You know, we're always straddling having a meeting at a time when we have enough data, but also enough time to provide any manufacturing issues related to recommendations. Our colleagues in the WHO, of course, met earlier, and I'm just wondering if there would be recommendations around the ideal perfect timing of meetings if this information that happens today or the discussions that happen today are something that should occur right now. This is ideal, or at a different point in time. That's my first question. My second question is, you've probably heard discussions starting last year about the difficulty in the H3N2, thinking about changing recommendation to think about having 2 different strains for that. So going to a different quadrivalent, and what are the sort of thoughts around that?
So that's a great question.
In the timing of the meeting, we would not want to see the meeting any later. It's really important that we get confirmation of the strains, fully acknowledging that we need sufficient data to make that recommendation as well. But for the current vaccine manufacturers, this timing of the meeting, it's important that it doesn't get pushed out any later. In response to the H3N2 question, yes, the H3N2 viruses are problematic. I know there has been discussion. I mean, even when we were talking about, even before we took the B Yamagata out of the vaccine, there was talk about the potential for, I think, Dr. Monto, you referred to it as maintaining the antigenic weight of the vaccine and, you know, adding.
I don't remember, but that was a good idea. and I thought we could do it a lot more seamlessly than we did.
Yeah. Well, of course, you know, I mean, first of all, we'd have to go through clinical trials.
Yes.
We'd have to get regulatory approval. And there are also some technical challenges. So, you know, if 2 H3s are very different, then it might be an obvious decision to have 2 H3s. But if there's not such a big difference, do we still put the 2 H3s in? You know, we may see interference between the 2 viruses. Certainly, the potency testing poses some technical challenges which have to be overcome. So, you know, we agree it's an interesting idea. Some preliminary work has been done, but there are a number of hurdles for us to get over before we can think about this new quadrivalent.
So this is a great point. Basically, the question had to do, that second question had to do with whether or not it would be a good idea to actually put 2 different H3N2 versions into the flu vaccine. And what Dr. Taylor was saying is probably it would, and people have been talking about this for a long time, but there are a lot of issues with doing that. The main issues being that type of vaccine has never been approved. So even though there's previously been a quadrivalent flu vaccine, it was with influenza B Yamagata and not 2 different kinds of influenza A, H3N2. H3N2 is usually thought to be more pathogenic than H1N1. So it does make sense to have broader protection against H3N2. But because a vaccine with 2 H3N2 components has never been made before, it would have to go through a full FDA approval process. What they're not saying is that with the current FDA leadership, and even though Vinay Prasad is leaving as the director of CBER, the vaccine regulator, you know, the FDA commissioner believes that HIV came from a lab and that Lyme disease is a Cold War bioweapon developed by a Nazi war criminal who escaped from Nuremberg and moved to Plum Island, where they have a animal disease facility. These are not the people who should be making decisions about whether or not having a quadrivalent vaccine with 2 H3N2 components is a good idea. But that, unfortunately, is the climate we're in. So I think Beverly Taylor's answer there was that good idea. Who knows? We'll see. Probably not super encouraging.
Questions from the group before I thank Dr. Taylor again for a very instructive presentation. Seeing none, thank you again.
Thank you very much.
That was a great
And
presentation.
now we have some time available for a more general discussion.
Here we
among the
go.
group. So let's have first some discussion in general, since we've talked about a lot of different things, and then focus on the strains
I'm
themselves
going to let them get the
before we
discussion
lead into
started.
the vote.
Get myself a
So,
fresh glass of
General
water.
discussion.
Jessica should be here
from
any minute.
the members, those who have votes, let's say. Dr. Durbin.
I just wanted to say I thought the presentations today were incredibly informative and really, really helpful to discussion. I don't have I don't have any questions regarding what was presented. I think the follow-up questions were informative. And I just want to thank all of the presenters for really a wonderful presentation of data that really, I think, makes our decision-making a lot easier. Thank you.
Dr. El-Sali.
Thank you, Arnold. My question is, well, we can ask questions to the presenters in general now, right?
Yes.
Okay, so it's between a comment and a question, which is vaccine coverage. I brought that up earlier in the day to understand sort of the nuances of the VE data. And then later in the day, the industry representative indicated that they are supplying less and less vaccine to the American market, which means probably less and less uptake of the vaccine. So I am wondering if a general overview of vaccine uptake can be incorporated in the deliberations we have here without, of course, veering into the details.
But it's my understanding it's not that the vaccine is not available for the U.S. market. It's that there has not been the demand.
Okay. Exactly. And
for the U.S. market. And I'm not sure if this comes under the purview of this committee, although I'd be very happy to hear from either the FDA or the CDC folks how this might be, how we might have a role. It's certainly whatever the efficacy if people aren't getting the vaccine, we're not going to see protection. Dr.
I wasn't looking for a role for this committee, which is more into the licensing end of the equation, just understanding the epi data in general. That's what I was.
Well,
yeah.
Dr. Grosko.
Hi, thanks for that question. I similarly, I can't really answer the question about the role of the committee and using it for decision making, but certainly if it's thought that having some kind of an update about that might be thought helpful in terms of interpreting the VA data, and if that's requested, we can do that. One thing to consider, a couple of things to consider. Those data, similarly to the VE data, are sort of collected throughout the season. They're not finalized usually until the fall. So we would have to ask our colleagues in the appropriate division, to be able to do that. But that's something we can pursue if the committee wants it. Also, coverage data tends to come from various surveys, and there might be differences in the sampling, which might not, at least for the CDC data, necessarily coincide with the VE data sites. So there would be caveats, of course, but it's something that, should the committee want it, we can of course pursue being able to do that. It's not data that our division produces; it's a sister division, but that's something we can look into if it's desired. Thank you.
I think it might be useful for future deliberations. Clearly, we can't do a whole lot right now. Given it was last year's data that were this past season's data that we'd be looking at. Dr. Gans.
Thanks so much. I did want to continue on this concept because I do feel that some of the role of this committee, given that we are presenting data that is wildly thought about within the general population, that we we should state some of the other efficacies that vaccines have, because it does relate to what this committee does. If there's low uptake in these markets, there's going to be a disincentive for our colleagues within industry to make vaccines. And so I think it is actually relevant to the work that this committee does, and in particular in the discussions that we have. One of the ways in which I think there's been some misconception is when we rely so heavily on vaccine efficacy, because we need that for our decision making, but I'm not sure that the general public understands when we use words like low efficacy, not as efficacious, and language such as that. So I do think that we're using data just to look at what is the concordance between a strain and what should be put into the vaccine, and how immunity to that in particular very myopically is associated. I do think we need to have the broader discussions about what are not only the sort of value to individuals and the value to society for having vaccines that do prevent, particularly, and I'll talk from the pediatric standpoint, hospitalizations in healthy people who don't otherwise have a reason to go to the hospital. The other part of that, of course, is a quality type of evaluation. And then if we want to talk about sort of broader impacts of what vaccine versus natural immunity could do, because I think there's also a conception in the public sphere that having natural immunity is better than having vaccine immunity, and not understanding some of the other adversity that could come. So for instance, I mean, I don't know what is compelling to people, but talking about like cardiovascular effects of having these viral illnesses. So I do think that there is some responsibility of this committee to think more broadly about what it is that we're considering in terms of the vaccination. Having said that, I mean, I think that the data that is in front of us is very compelling for the recommendations that are facing us today. The only additional conversation that I think this committee needs to have, and we've been having it, but to your point, Dr. Monta, we've been having it, and we're not sure what further discussion or sort of pressure needs to be put on having a discussion about how we can actually prepare vaccines in a timely way that can use the data that we're all getting and be able to kind of move more nimbly between the data and a recommendation. So we've been talking about an H3N2 that really needs to be expanded, probably to give us better vaccine efficacy if we really want to talk about it from that standpoint. And I think just like going from a quadrivalent to trivalent, there was some frustration in this group to have to discuss it for years before things happen. So I would urge our colleagues, and maybe it's us as responsible group to urge more forcefully to have those studies available so that we can be more nimble in what we would like to do.
I just wanted to cheer during that entire thing.
Pandemic shifted the attention not only of the public, but of the scientific community away from influenza, because the same people very largely that were working on influenza went to work on the prevention of SARS-CoV-2 and development of vaccines. And this happened just when the approach to a broadly based influenza vaccine was really moving forward. And really what we have to do is re-examine where we are. And I'm not sure where that ought to be done, because things have changed in many respects.
Boy, you can say that again, Arnold.
Since before the pandemic and more recently. But we really need to focus.
Yeah, like in the last year and 2 months.
The broad picture. And we've talked about the two H3N2 strains in the vaccine, but there are a lot of other approaches, and they ought to be looked at comprehensively. And to also in a way to let the public know how we are looking to make a good vaccine a better vaccine. After that preaching to the converted, any other comments before we look at any comments about the specific strains in the vaccine?
Boy, that was a great discussion, and Jessica and I are going to unpack that one in more detail later. Dr. Gans is a goddess.
The other panelists have also said, I just really wanted to thank all of the presenters today. I thought this was an excellent session, very clear, very comprehensive. I think as it relates to the strains and what we're talking about, I think, I think the entire set of presentations laid out very clearly in a way that leads me to support the proposed recommendations. I didn't see any indication that we should move into a different direction. So that's where I stand on this. Thank you.
Okay, any further comments about the strains? And I'm not going to ask people who don't to explain their votes, because generally we are all in agreement with the recommendations that are put in front of us. Any further comments?
You know, I'm going to disagree with Dr. Monto on the need to do that. I was really glad to hear Captain Meyer say that she was persuaded by the evidence, because that's what's going out of the federal government right now, is making evidence-based health policy.
So early in the afternoon?
It's great to see somebody in the public health service defend evidence-based medicine while in uniform.
Are you asking the committee members or Cicely?
I'm asking both.
I can vote early.
I think we're ready to vote. I just want.
Okay.
Perfectly fine.
Okay.
Action starts now.
To the voting portion of the meeting, and the questions before the committee today. There are two questions. They are both voting questions. The voting members will use the Zoom platform to submit their votes for the meeting. If you're not a voting member, you will be moved to a breakout room while we conduct the vote.
All right. So what's going on right now is just explaining the voting process and how it works over Zoom. I'm just going to explain very quickly during this why they actually vote. So much like the ACIP, the VRBPAC makes recommendations, except they make them to the FDA for what they should approve or not. So for the purpose of this meeting is to have this vote in which they vote on the recommendations that they're going to make. At issue is which vaccine, which viruses will be included in both the egg-based and the cell and recombinant-based influenza vaccines. Which viruses are they going to contain, more or less? So, you know, we're hearing all about the details of the different voting processes and how the votes are tallied. Long story short, it's a very typical democratic process in which, you know, they go through all of the different voting members. There are some non-voting members on the VRBPAC. The VRBPAC needs to have, I think, 15 members in order to vote. So they've brought in some people who are sort of veterans from the VRBPAC, from VRBPAC's past, including Dr. Monto here. And now he's going to call for the vote. So I'm going to shut up.
Hey, does the committee recommend a 2026-27 formulation for egg-based influenza virus vaccines in the United States that contain the following virus strains? And A, Missouri 11 2025 H1N1
Currently circulating
pandemic
H1N1 seasonal
0609
virus,
like
derivative of the
virus,
2009 pandemic.
and A. Darwin 1454 2025
H3N2 subclade
like
K.
virus, and a B. Tokyo
Influenza
EIS
B component.
13175 2025 B. Victoria lineage like virus.
And these are the viruses that are good for growing in eggs.
That is the first question. Are we voting now for number 2 as well?
No, we're going to do
Yeah,
the two.
that's what I thought. We're doing them in sequence. So we vote for 1 right now.
Egg vote up first.
No, we're going to vote for 1 and 2, so you can read both questions.
We're voting for the
Never
cell and
mind.
recombinant as well. Okay.
Yes.
Does the committee recommend a 2026-27 formulation for cell and recombinant-based influenza vaccines in the United States that contain the following virus strains: an A. Missouri 11 2025 H1N1 pandemic 09 like virus, an A. Darwin 1415 2025 H3N2 like virus, and a B. Pennsylvania 2025 B. Victoria
So basically,
lineage.
the only one that's different is this virus, and it's just
Thank you,
B
Dr.
virus that's
Monto.
better suited for
voting will
growing in
now
cell
begin.
culture. Here we go. Voting in progress. Are you on the edge of your seat? Because I am. I'm not really on the edge of my seat. I have a feeling that everybody's going to vote to accept the WHO recommendations. But this is a little different than the ACIP in that the ACIP, when they vote, they do it verbally. And it's sort of more like a city council meeting, or I don't know, a student government meeting. There's, nomination of this recommendation and then second it, and then parliamentary procedure and all that. And then they vote verbally, and sometimes will verbally declare if they abstain for one reason or another. Here it's a little different because they use I think a Zoom-based mechanism to actually register their votes. It has to be a little different for the ACIP because that meeting is occurring in person, and it looks like for this meeting, all of the VRBPAC members are joining virtually over a Zoom channel rather than actually going to, well, in this case, Silver Spring, Maryland. For ACIP, it's Atlanta. But they're not always all there in person. And today it looks like most of them are not actually meeting in person. So I guess that this is the quiet period where there is, Zoom voting going on. This is not a very unexpected outcome. And in some ways, this is sort of a very anticlimactic meeting. What I found the most surprising about this meeting is that it even happened. Last year, VRBPAC was canceled, really without any sort of explanation. And a group of unnamed agency officials from, I believe, the then called the Department of Defense, and several agencies from within HHS all met on a Friday night and basically just decided to accept the WHO recommendations. Normally, that's what happens every year. The only time that there might be a difference is if, U.S. surveillance data that is more recent even than what has been put into the WHO GISRA's system is, forthcoming, or they're able to analyze more data that would inform them that for the US, a different strain would be better. Sometimes in the manufacturing process as well. Another virus strain might be better. The viruses themselves are made using what's called a reverse genetic system. So it basically is taking a bunch of DNA that encodes for the virus and putting it into cells and getting those cells to make virus. Sometimes with the egg-based or the cell-based systems, these aren't optimal. And so you end up making other sort of reverse genetic systems that will still have the CVV antigens, the H and sometimes the N, but will have other components of the virus that will make them grow better in one system or another. So those are all various reasons why they might not accept the WHO recommendations. But importantly, historically in the past, all of these recommendations, they go through this process every time because all of these recommendations have to be evidence-based. So they don't just have 1 year where somebody on the VRBPAC is like, you know what? I don't like the look of the WHO's recommendations. I don't like that particular strain. Missouri pandemic, 2009, can just, launch itself into the sun. I don't care about it. I want to use this other CVV. People don't just say that. And historically, for vaccine regulation, any kind of drug or medication regulation, you don't want people who are making decisions based on their gut instincts. You want people who are making decisions based on what the evidence shows and based on a standard that everybody agrees on for making an evidence-based decision, especially when it's something as important as a decision that's going to be translated into a drug approval. So I don't think that people, recognize, because we've taken it for granted, that the FDA exists and it does its job in an evidence-based, scientific way, and that its goals are to bring drugs to the market that are safe and that actually work. And that's why the FDA was started. The FDA was started essentially because there were quacks selling literal snake oil and saying that it was medicine, and it was hurting people and it was causing them to suffer unnecessarily. And it was, in some cases, causing people to be poisoned. I mean, there were a ton of problems with these unregulated medicines. And that's why the FDA has historically assembled advisory committees or AdComs like the VRBPAC. Because even though we do know very well how these flu vaccines work, it is still important to make the decision that affects so many people's lives based on evidence and not like vibes or, oh, I just love a placebo-controlled trial, so therefore I'm going to insist on that because I want it. That was the previous policy of the outgoing CBER director, Vinay Prasad. That appears to be sort of the policy of the FDA commissioner, except the FDA commissioner also has, Marty McCary, has instituted this voucher system, which basically allows a company to pay their way to the front of the line and provides the Trump administration and the MAHA movement a mechanism to essentially shake down the manufacturers. However, unfortunately, as we just heard from Beverly Taylor, the manufacturing process is extremely complicated and it operates with an extremely unforgiving timeline. So if, as Beverly Taylor was asked from Securus, one of the biggest flu vaccine manufacturers, this meeting, the VRBPAC meeting that's happening today, cannot happen any later than this. If it were to happen any later than this, there would be no flu vaccines available in time for flu season. That's why the FDA's new policy, or I guess it's a year old now, but their policy of insisting on quote unquote gold standard science that has never really been defined, but does seem to involve a placebo-controlled trial for everything. That is not possible with the flu vaccine. And the reason for that, there are several reasons for that, but one of them is that you simply can't conduct a phase 3 clinical trial, placebo-controlled or not, within the time frame that we have to make a decision about which strains we're going to include in these vaccines, and when we need to actually start making these vaccines to make sure that we have enough of them to actually vaccinate people. And as the committee was discussing, as I discussed at the very beginning a little bit with Saskia, these vaccines, do work. And as a couple of the VRBPAC members pointed out in their comments, they really work most of all by keeping people out of the hospital, by reducing the strain on our healthcare system, and by keeping more people healthier, even if we don't keep them from becoming infected with these seasonal influenza viruses that we've tried and we keep trying, but we haven't been able to get rid of them. And, it's not looking like we will in the foreseeable future. And as a virologist, I'm not even sure that it's possible to get rid of influenza viruses. They infect so many different species, and there's so many of them that also don't infect humans, but could. And so, it is really, really important that we use whatever piece of data we can when we're designing these immunization programs, because they aren't perfect, but they really do work at population scale, even if they don't prevent individual people from getting infected with influenza virus and perhaps having some symptoms. In general, these vaccines work tremendously well. And one of the things I was so encouraged to see with regard to this meeting is just that the VRBPAC members in that last session were just so thoughtful about what the role of the VRBPAC is. It's not lost on me that it's probably in the back of all of their minds that the VRBPAC could be canceled at any moment. Probably the better the VRBPAC does today in terms of adhering to an evidence-based, sensible, scientifically sound process, the more likely it is that the VRBPAC will never meet again, at least not with this particular composition of members. For those of you who were watching today and saw the question answer sessions with the members of the VRBPAC, it's completely obvious to anybody, whether you're an expert in the field or not, that these people have deep expertise and that they know what they're talking about. And in fact, most of them have been working on influenza vaccines, or treating influenza patients, both pediatric and adult, for decades. There are literally centuries of experience working with influenza viruses and influenza vaccines on this committee, which makes for a pretty boring advisory committee meeting because, again, flu vaccines work. We've had them for a long time. This process has been historically fairly routine, where you heard references to long and tedious sounding debates, and oh boy, they were. I wasn't even going to VRBPAC meetings regularly, but I'm in the field enough to know that 10 or 15 years ago, people were having conversations about adding additional H3N2 components to the vaccine and things like that. And, it has gotten very tedious because the flu vaccines actually work so well that really the main problem has been these bureaucratic procedures needed to update the vaccines or to incorporate new technology. What I thought was not said that explicitly, but was definitely the topic of that last discussion section, is that the FDA is not reliable to make decisions based on evidence at all anymore. The FDA commissioner is actually is spreading conspiracy theories. The FDA commissioner is having anonymous press conferences in which it's like, don't tell them it's me, says a guy who looks and sounds exactly like Marty McCary. And that's the FDA that we're dealing with now. So I think that this VRBPAC meeting, in a way, is kind of like it may be a last hurrah. It was just wonderful to see them discussing that stuff. Now, it is just wonderful also to see that this is back.
Thank you, everyone, for your
and my co-host is here. Finally, I'm letting
the
Jessica
voting
into the
has
room,
now
quieting down
closed
so we can
and
hear
is
the
now
vote
complete.
results. Then we're going to chat.
The voting results will be displayed. Okay. My 53-year-old eyes can barely see that, but I can tell that we have, for question 1, we have a unanimous vote of 7 yeses and 0 noes, 0 abstains. For question 2, we also have 7 yeses, 0 noes, and 0 abstain.
I can also barely read.
Okay, Dr. Montel, if you would like to just go down the line and have everybody state their name for the record and how they voted.
Okay, it seems a bit superfluous to do it, but I guess we need to do it for the record. Does everybody see the names that are on the left, which started with Dr. El Sali? So Dr. El Sali, you go first.
To give comments?
No, this is to announce how you voted. We're doing it differently.
Oh, I announced. Okay. I thought
And you can do it for both. We don't have to go down twice.
Okay, all right. I voted yes for questions 1 and questions 2.
And so did I. Voted yes for, and Dr. Gans.
Dr. Haley Gans, I voted yes for both questions 1 and 2.
Dr. Meyer.
Hi, I voted yes for questions 1 and 2 as well.
Dr. Berger.
I also voted yes for both questions 1 and 2.
Dr. Durbin.
I voted yes for questions 1 and 2.
Yeah, I guess we don't have to listen to them all
Dr.
say, I voted
Perlman.
yes.
I voted yes for questions 1 and 2.
Okay, I guess we're all in it for the record.
Okay.
Now, we have a couple of options. And if anybody wants to explain their votes, they can do so now. It is not required. And then if there are any further comments, this is your time to raise your hand and ask and make a comment.
So the reason I'm being kind of quiet here is that I do anticipate that we might get a lot of sort of collegially phrased academic statements of resistance
Seeing
here.
no hands raised, I'll just
But perhaps
say
not.
that we've had a very efficient meeting in a very complicated year where we were reminded about how difficult strain selection is, that it's a little bit of science, a little bit of luck in coming up with the right strains for the vaccine, which is why we really need a broader vaccine so that we don't have some of the issues that we've been discussing all day today. Other than that, I just want to thank everyone who has put this meeting together and, more importantly, done a wonderful job gathering the data, doing the analysis, and doing it under sometimes difficult circumstances. So again, thank you all. And I'd like to turn the meeting over to Dr. Reese, who will be inviting Dr. Kaslow to speak, or anyone else who from FDA, from CBER, who would like to add his comments.
Blink twice if you need help.
Okay, well, I was looking for Dr. Kaslow. I don't see him join, but I'll see if he wants
that's what I was wondering when you end early.
There you go.
There he is.
Thank you.
There he is.
Thank you. There I am. Thank you, Chair Monto.
So this
let
is
me
who
start with
Vinay
a
sent
few
in his
brief
place.
reflections before ending with some thank yous.
He's still
So
nominally
today,
the
VRBPAC
CBER
convened
director.
for the 190.
But, you know, I guess he didn't really want to pop in and embarrass himself. Sorry, I think the stream froze. I'm not sure that Dr. Kaslow is going to say anything that interesting.
Is there,
He's back.
is the sound just cut out for me?
No,
No,
that's Dr.
I think
Monto.
they're working on it.
Okay.
Oh boy. You gotta love when the Zoom fails during the closing remarks.
Of seasonal influenza vaccines. As I think you all noted, today's VRBPAC discussion went well beyond the voting questions. It brought into the public discourse emerging issues that warrant consideration and being better prepared for future respiratory seasons. It also highlighted the scientific and technical challenges of always having to chase the rapid evolution of these, I think you called it, tricky viruses. As Dr. Weir pointed out, the way forward starts with defining and getting to a shared understanding of the unmet medical need, and then thoughtfully and comprehensively framing the problems to be solved. Just like your prior discussions in 2023 and 2024 on the transition from quadrivalent to trivalent vaccines, and your sage advice last October to keep a close eye on the influenza B Austria-like virus that hadn't changed for a number of years, your discussion today is yet another call to action for even more effective seasonal influenza vaccines. So thank you again for another robust, evidence-driven discussion to drive improving public health against a very serious and life-threatening pathogen. So I'll end by first thanking the open public hearing speakers, the private citizens and organizations that posted comments to the docket. The
Save
office
America
hears you
movement
and considers your points in our internal discussions. Let me also thank all of the invited presenters for their outstanding interventions today. You drove a robust public discussion, noting both the strengths and the gaps in the evidence. And the setting for those presentations would not have occurred without a large team here at FDA to host yet another flawless VRBPAC. So thank you, FDA Advisory Committee staff, our AV staff, and my colleagues in OVRR. And finally, a sincere thanks to today's VRBPAC members for your courage in the name of public service. Your service is vital for enhancing public trust. And I'll end by saying, we look forward to a 192nd convening of VRBPAC. So with that, I'll turn it back to our DFO. Thank you.
Thank you, Dr. Caslow. And thank you also, Dr. Monto, committee members and speakers for your time and contribution.
All right. Sorry, Dr. Reese. We're done.
The deal was done. The votes are in.
Yep, the votes are in. Unanimous on both. One was first vote. You came in right at the end when I was like just chatting my way through the Zoom voting procedure. Oh boy, I've been doing a lot of just talking to myself. Also, I was saved from talking to myself because I couldn't figure out how to make the sound work on any YouTube video. And while I was talking to Saskia, I accidentally hit the back button on my browser and booted myself out of my own production studio. So, you know, there's a few technical kinks that need to be worked out for next time. But it went pretty well. It was a pretty boring meeting. And, you know, so I watched the morning part.
Yeah,
even though I didn't.
Boring though. Right.
It was boring just because, like, for anybody watching that, and back in the day, like when the FDA worked, and I also wasn't in charge of a vaccine journal where I actually had to follow this stuff professionally, like I didn't watch these meetings. Like I'm familiar with this data. Yep. It doesn't really change every year. You know, the effectiveness changes from one year to the next. And the strain selection, like, you know, it's a data-informed crapshoot, as we saw this year with subclade K. But you know what? I mean, it's really interesting to watch the process when you're like, I may never see another VRBPAC meeting so boring and evidence focused again. And it's interesting to watch it through that lens because they are so thorough. Like they really want to make sure that the WHO recommendations are going to be right for the US population. And I mean, it was so like such like data dense presentations that were provided. That, you know, it's just really kind of sad to see like that we're not going to have as many boring meetings again, because the more I think this meeting like accomplishes what it's supposed to do, which is advise the FDA on what they should approve based on evidence, the more, you know, they run the risk of being permanently canceled or replaced with a bunch of insane people, or, you know, the stuff that's already been done throughout HHS.
Right. So let's talk about what we're on.
You know, what I thought was the most interesting part of the meeting, and I know you're just walking into this, so I guess I'm giving you a recap, but I'm kind of also giving everybody else a recap. So the meeting was boring. They went with the WHO recommendations. The recommendations, there were two. One is for egg-based vaccines, 3 candidate vaccine viruses, which are the collection of viruses used for vaccine manufacturing that these WHO collaborating centers around the world maintain. So it was basically just recommending the 3 of those. So H3N2 component, the H1N1 component, and then the influenza B Victoria lineage component. There was a big, long discussion about that because the flu B has changed antigenically and it hasn't changed historically very much. So a lot of, as you would call it, inside baseball flu stuff. But a really good, you know, thorough discussion. And I think, you know, I feel pretty confident that sure enough, like the WHO recommendations were as good as they could be with the data that we have. But here's what I thought was the most interesting thing about the whole thing was the discussion at the end. Now, not a lot of people were chiming in, but the people who did were extremely thoughtful. And so Haley Gans in particular made this comment that I just like, I was silent through the whole thing because I didn't want to like talk over her. It was really amazing. But the whole time I was like, yes, like, I mean, it was just really good. And it was delivered in like a pitch perfect, like academic, I'm on an advisory committee tone. But she basically said that the role of, because there was some discussion about what the role of VRBPAC is. Like, should VRBPAC address vaccine hesitancy or not even vaccine hesitancy? They referred to it, I think, as like not low vaccine uptake and things like that. And what Dr. Gans said was essentially that we need to, it's all stuff that you and I have talked about, basically, that framing vaccines in the context of efficacy data is good for making policy decisions based on evidence, but it is not good at communicating the benefits of taking vaccines to the public. And the whole debate was like, is that the VRBPAC's job to worry about whether the public has confidence in vaccines, because they're not making recommendations to the public, they're making them to the FDA. But her argument was that absolutely that is the VRBPAC's role. And it was very interesting to listen to them talk about this. Arnold Monto said some comments to that effect as well in response to this. It was very interesting how they weren't directly saying like, oh, it's a bunch of anti-vaxxers running the FDA and like Marty McCary's taking, you know, payoffs, like basically. And then up until, you know, recently, Vinay has been just trampling all over rare disease drugs and vaccines and all mRNA vaccines and all of Moderna. And, you know, all this chaos has been happening. Tracy Beth Hogue is dumpster diving in VAERS and finding made up dead kids that got vaccinated. You know, it's FDA is a complete and total mess and it's not capable of regulating drugs effectively. But nobody actually ever said that. They just said that our role is to make sure that FDA has all of this information that's based on evidence. And we have a lot of evidence about vaccine uptake. we need to start incorporating that into our recommendations, because ultimately we're not going to have as much data because CDC has been so depleted and we're not participating as fully, and we're not capable of doing the same level of surveillance that we used to do. And there was a whole other discussion immediately prior to this from Beverly Taylor from CSL Seqirus, which is one of the US's largest flu vaccine manufacturers.
Right.
And I mean, it was fantastic because she went over all of like the extreme time constraints for having vaccines ready. And that's directly relevant. I was just jabbering to myself and, you know, whoever's watching. I have no idea, by the way, who's watching because I can't check like my other platforms from this streaming platform. So hi, everybody. And I'm sorry if I didn't answer your questions, and I'm going to do better next time. But, you know, I was just kind of going off on this because it's incredibly important. If the FDA's policy going forward is that it doesn't matter what the evidence shows, it matters that there has to be a RCT on everything. There has to be a placebo control arm, you know, all this stuff that would basically have the effect of making flu vaccines completely not manufacturable. I mean,
yeah.
There was a whole discussion, and it's really challenging to explain some of these things to people, because it's like, if it takes, you know, so much time, why don't you just start before? And it's like, because we don't have enough, it's such a fine balance where there's like this 3-week period really where there's enough data to accurately do strain selection and then get the information to the manufacturers and get the CVVs to the manufacturers. I mean, that's the thing. They also can't make vaccines with candidate vaccine viruses if they don't actually have the candidate vaccine viruses. So Beverly Taylor was also talking about the issues with benefit sharing policy, IP, all of the issues surrounding that, and then the further complexity of all of these different bureaucratic instruments that are trying to regulate this, and they might be in conflict with each other. I mean, it's just incredibly complex. And so what the conclusion that I think Haley, the argument that she was ultimately making, and she also brought up the example of the public not understanding the difference between vaccine-acquired immunity and natural immunity, which the FDA commissioner also is a big believer in. She was talking about how essentially, you know, we keep thinking about evidence-based vaccine policy and like trying to make our role as narrow as possible. But what is the role? It's to advise the FDA on what's the right thing to do, and to make vaccines as available as they can be for the American people if the evidence shows that they offer a significant benefit. And I just really thought it was so nice to hear. I mean, you know, it brings a little tear to my eye to see all of my colleagues having this discussion about, they know like that this could be the last time that they meet, or that anything like the VRBPAC meets at FDA. They certainly know the condition of FDA in terms of its regulatory capabilities and the competence of its leadership. And it was just really nice to hear people having a discussion, even though it was in very collegial, polite academic language, that was addressing some of these things and thinking about how they would take a proactive role in making sure that evidence-based policy and that vaccination remains a priority within the American health system.
Yeah. You know, the communication side of it becomes very clear to me, as flu vaccines really are a combination of evidence and luck. And that makes me feel very uncomfortable, because like, what do you mean luck? It has to be like rigorous science. We're like, it is rigorous science. How quickly we have to react to Southern Hemisphere data and probability data and hospitalization data that has a massive lag, and acute outcome data that also has massive lag, and be as active and reactive as possible to prepare for our incoming flu season. There is a bit of like a gamble there. Now, it's not a gamble with people's health. It's a gamble in efficacy. The safety is not what we're gambling with at all. And I think that that's, people don't understand the why behind why we do flu vaccine. I mean, the fact that there are people today who still believe you can get flu from the flu vaccine, or that flu vaccine is not your get out of card, you can go live in flu season and not get flu. I've still got a lot of people in my DMs being like, the only time I got a vaccine, I got the flu, and you can have the other. Very less teachable. But what I try to remind folks, help them aspect of, I don't want people as scientific as it really is, actually to a degree, is that flu vaccines, since the flu vaccines were invented, are intended to do things, to get you out of a hospital and to keep you out of a grave. And they're damn good at doing both of those things. Know this, and this goes back to what Casey means was incapable or unrecognized. Years and years and years, effective is what we get from the real world, not from clinical trials. We don't have time to every year. Effectiveness, real world tells us that flu vaccines do really good of keeping people outcomes and outside of graves. And so it concerns me that kind of like risk benefit message is not what we're screaming from the rooftops, because now this idea of relative risk is now kind of being related who don't really understand risk. And I would like for that to, I hope that this is not the meets. I hope this is the last time we're not considering modifying or updating flu strains based on the data that's shared on the show, because out of the flu vaccine is essential. We used to lose more every single year, including healthy folks and young folks, to the flu for no reason. We know we have something that is doing those two things: keep you out of a hospital and keep you out of a grave.
Yeah, I could not agree more. Jessica, I just have to say, like, it might be my internet because I do live in Saskatoon, but you're a little bit kind of going in and out.
Choppy?
Yeah,
Oh,
a little
no.
choppy. I don't know if you want to try turning camera off and turning it back on or something, or even just logging back in. I don't know. It might be me too. I just, when we're, oh, she's gone. Okay. I'm going to address in a moment with Jessica, like what she was just talking about, because I couldn't agree more with what she said about the communication part of this being so important. What I found so striking about that VRBPAC meeting was it got really spicy there at the end with Haley Gantz's comment. Hi, Jessica, you're back. I was just going to say that I wasn't going to address what we were just chatting about until you got back. Let me put you back on screen. I promise I'm going to be a lot better at this production thing after this.
You're fine.
Actually. Yeah, that's what I figured. This is kind of a nice meeting because, and I think tomorrow, tomorrow, Jess and I, we said we're going to have a weekly show, and we sure are. So tomorrow is our weekly Friday afternoon episode of Taking Shots, and we will unpack this in a more structured way once Jessica's also had some time to digest the highlights. But, what I just found so striking about this is that what you just said about communication, communicating the risks and the benefits. We have discussed that we have a early career male colleague who is not always so great to women. And at one point, he and I had a pretty acrimonious debate about what is the purpose of vaccines. And he was running with the line, probably because he actually doesn't work on vaccines. But he was running with the line that vaccines only work essentially if they prevent infection, if they provide sterilizing immunity. Why don't we have sterilizing immunity? Why didn't we invent vaccines that would have sterilizing immunity in the first place? Well, the first
vaccines without telling me.
The first vaccine was a virus, except Edward Jenner, who gets credit for it, he didn't really discover it, but he's the one who gets credit for it, named it Vaccinia because that's he was wild about Latin, and that means cowpox because it's cowpox virus, or it's actually Vaccinia virus, and cowpox is something different, but virology thing not to get into. Anyways, it's a virus. He didn't know what a virus was. Germ theory wouldn't be invented for another 75, 80 years. He called it a virus because, like I said, he was wild about Latin, and virus means poison in Latin. So he was calling it a virus, but he actually meant it's a poisonous, like toxic substance. He didn't know that it was an orthopox virus. The purpose of that was not to prevent infection. Like Edward Jenner had no idea what an infectious disease was. Like that's what germ theory means. Germ theory means that you think that pathogens cause disease. I don't know if he was a miasma guy or what he was, but he didn't know about viruses and he didn't know about pathogens. So, Robert Koch was a baby, like when he did this. So, vaccines were not made to stop transmission. They were made or infection of pathogens. They were made to stop disease. They were made to stop new cases of that pathogen. And, I've had so many people, usually, again, overconfident men who don't work on vaccines, explain to me over and over that this is why the public doesn't trust vaccination. And no, I mean, this is what the VRBPAC was talking about. When we talk about something having, 62% efficacy or effectiveness, I mean, we're talking about prevention of influenza disease, cases that would be recognized clinically. They might be diagnosed in using molecular tests, but they are cases that were found because of clinical symptoms. That's not preventing infection. That's a readout that is prevention of disease. And flu vaccines do work very well for that. You might get infected with the virus, but you don't get very sick. Like, as far as I'm concerned, whatever. I can't escape getting infected by the virus, but if I can make it less likely that I'm going to end up in the hospital, count me in. And like you were saying, you hear the same things over and over again. Flu vaccines cause the flu. I've heard that one so many times and it just makes me want to scream. I was actually delighted when Leslie Manookian, who is like a children's health defense anti-vaxxer, got into my replies on Twitter one time and actually said that she had Guillain-Barré syndrome. And I was like, thank God, it's a refreshing change from it gave me the flu. She doesn't have Guillain-Barré syndrome, by the way. I don't think. But, we hear these things over and over. And one of them is that flu vaccines don't work. Respiratory vaccines don't work. You still get COVID. They overpromise and underdelivered. No, these vaccines work very well. Like you said, they keep you out of the hospital and they keep you out of the grave. Like, what else do you want? I mean, I'd love to have a vaccine that prevented it. But in the absence of that, I'll take continuing to live as a better option.
Yeah. I mean, it's why I continue to do the work that I do, is the message is still ubiquitous enough. It's not, it hasn't penetrated minds where the old takes and the understandings of how flu science works is well known. And it is unfortunate because I think back to some of the betrayal that a lot of people feel about the COVID-19 pandemic, because people felt deceived in what they thought the COVID-19 vaccine was going to do, because there was talk about it's real business, it's going to bring you back to your family, it's going to bring you back to your weddings, funerals, and everything that you had to defer. We're like, if enough people got vaccinated, we could reduce transmission. But this is, Angie, where it drove me crazy, because we were seeing even messages from the White House, things like, if you got the vaccine, you could take the mask off. That's not how public works. It's layers and layers of harm reduction, and vaccines are of harm reduction. It's not elimination. And I think that's again where the messages get lost. We cannot eliminate risk. Being alive is risky. There's risk in everything. There's risk in drinking water. I can choke, I just feel exactly. So this is really, time in public understand, because that a whole movement trying to downplay risk of very serious consequences, including flu, measles, etc., even COVID. And we're trying to say, let's talk about risk in a scientific way, and sense that you're actually evaluating your risk in their way. Because viruses are a terrible way to get immunity. Vaccines are a much low risk way.
Yeah, I mean, this is what I've never understood. This idea that like measles, which I've never had, obviously, because both of my parents had measles. So they didn't want me to get measles either. They both remember measles.
Right.
Everything I've ever seen about measles looks like it sucks. Now that I've actually seen what a measles outbreak looks like in the US, it looks like it sucks. Like, I would hate it to have my kids going through that. I would hate it to go through that myself. Like, so I truly do not understand where people are coming from when they say things like, I'm glad my kid got measles. Like, it's good for them. It cures cancer. It causes like so much awesome immunity stuff. I mean, no, obviously, this is well documented at this point scientifically. Measles actually causes immune amnesia that makes you forget every pathogen you've ever encountered for a period of like 2 to 3 years. And so then you got to get all your vaccines again. And they have actually done, and you would know this better than me because I'm not an epidemiologist and you are, but they have actually done epi studies now to show that populations where there's been a big measles outbreak are much more vulnerable to other infectious diseases.
That's right.
The non-specific effects of pathogen infection. But
That's
it
right.
is always, to your point, lower risk to take a vaccine that's been approved and is known to be safe than it is to get infected with the actual pathogen. And that's literally what we have vaccines for. It's so that you can develop immunity to the pathogen without the part where you get sick. And even if it doesn't kill me, even if like the risk of something like SSPE is low, and although I did just see a study, which admittedly I haven't read in full, that says that SSP has actually been grossly underestimated. So this is a type of encephalitis that people can get years after a measles infection that is lethal in every case.
In every case.
In every case. Babies are particularly prone to it. So your baby gets measles. And this also applies. I mean, this affects people who are planning to vaccinate their children, but their baby is younger than a year old and cannot yet get the MMR vaccine because it is a live attenuated vaccine and it has risks on very, very young children under a year old. So unvaccinated babies who get measles are at risk for the rest of their life, essentially, of this lethal, incurable, progressive neurodegenerative disease. It's truly, truly horrible. And I just don't see how you can make the case that this is somehow better than vaccination. And the reason, of course, as we both know, Jess, is that because it's not, and people are lying about it.
Yeah. It's deeply, deeply sad that we are so okay with disability, death, and disease, and that we would be willing to subject our most vulnerable population, our children, to those things because we believe that the things that are intended to reduce the harm of disability, death, and disease are somehow greater. And, you know, in all of public health history, getting the disease is never the ideal strategy. It was strategies before we had interventions, and I'm thinking specifically about how we managed chickenpox. I am a generation that went to chickenpox parties. When there was chickenpox reported in everybody to school next day because that was the only way to just get it out, right? You're gonna have a bad week. Most people, you're gonna have a weekend of oatmeal bath. You're gonna be really itchy, but not everybody did okay with that outbreak, right? Like most people are okay, but some kids had severe outcomes, and that's all we had to do. That's all we had available to us to reduce that risk. My kids will never know that hell because my kid got the varicella vaccine, which has now been available for almost 8 years. Incredible. And I still have people asking me why that is considered a newer vaccine. I'm like, again, and Angie, I don't know if you see in quite a scientific line people draw with old school and school vaccines, very kind of being right that point where people make that very unscientific distinction. These are new and these are old. But think about the way we have reduced kids' harm is incredible. I mean, there are a number of European countries now that have been vaccinated for are now rethinking that decision because they realize these are very disruptive outbreaks. Some kids actually have some kids get really scarring and have horrible experiences because pox down the throat. I mean, it's miserable for some kids who get varicella. And so because of that, the UK being one among that are reversing those policies of not vaccinating against chickenpox because they realized virus-induced immunity is not greater than or better or less harmful vaccine-induced immunity.
Yeah, I mean, I couldn't agree more. And I think I'm a few years older than you. So I'm also in that chickenpox party generation. Let me tell you my story with that. So my mom had chickenpox when she was pregnant with me. Chickenpox is, she was 6 months pregnant with me. So chickenpox is a herpes virus, gamma herpes virus, varicella zoster virus. Right. It's named varicella zoster virus for the diseases that it causes. Varicella, I don't know if Edward Jenner named that one. He didn't. But, somebody who is very much of his own heart because it's a Latin name and it means chickenpox. Zoster, herpes zoster means shingles. So that's what the virus causes. When you get chickenpox, you get infected. You get pox all over your skin. It's called a lytic infection when a herpes virus is actually just replicating and making new viruses actively. That happens in the skin. But then those new progeny viruses go into the neurons and they migrate to the dorsal root ganglia, which is the bundle of nerves at the bottom of your spine. And then they go into latency, which means that the virus just sort of sits there in the cell. Mm-hmm. It doesn't do anything. It doesn't make more viruses, but its genome is there. It's a DNA virus, so it's very stable, and it can stay there for years and years and years. But then you can get a reactivation event. Usually this can be stress. This can be another infection. This can be a lot of different things. It's not always really well understood. But then you get shingles. You get herpes zoster. The reason why shingles goes around your midsection is because it goes through the nerves that it's in. So why would you want to expose yourself to that for the rest of your life? When you are older and you get varicella for the first time, like my mother did when she was also pregnant with me, so therefore immunocompromised, she was in the hospital. I mean, she was really, really sick because of that. Because it is a virus that infects ultimately your nerves. For whatever reason, in older people, they're more likely to have a severe case. So my mom did not want me to go through that. She's like, you need to get chickenpox when you're a toddler, so that you barely notice it and don't remember it because no vaccine. So she exposed me to chickenpox. I don't know if it was passive immunity, like that lasted a really long time or what, but I never got it. Fast forward to my doctor was like, maybe she's immune. Fast forward to I'm 17 years old, and I think that I have a hot tub rash from hanging out with my best friend at the time and smoking cigarettes in her hot tub. It wasn't a hot tub rash. It was chickenpox, in fact, and I was sent home from school immediately when literally I was sitting in class, and I was a senior in high school. And I started having pox appear on my face. I wasn't sick enough to be hospitalized, but I was really uncomfortable. And I remember I would only allow myself to scratch the ones on my head so that if I had a scar, it would be under my hair. I have one little one on my cheek, but it was terrible. And I had them, yes, in my throat, other places where the sun don't shine.
Yes.
Also inside my ears, that was uncomfortable. It was terrible. And I was really paranoid that I was going to have it on my eyes. And I was not a virologist, obviously, when I was 17, but later on, I was like, holy shit, am I glad I've never had an ocular herpes infection? Terrible blindness.
I mean, shingles in their eye.
Yeah. It's extremely painful. It just horrible. And why would you want that instead of a new vaccine that's actually like 30 years old. I mean,
Yeah.
the reason I didn't get the varicella vaccine was that when it did come out, and I want to say it was in like 1996, that was the year that I got chickenpox. And when it first came out, it was only recommended for very young children, which is also how vaccines often go. When the HPV vaccine came out, I was 26 years old and they were only giving it to 25-year-olds. And at that point, I'd already had an abnormal pap smear. So whatever, I got it the old fashioned way. But years later, when I was in my 40s, they expanded the age group because at that point they had enough data to say that it still benefits people, even people who've had HPV before, and even people who are older than the younger, generation that we're trying to prevent it in altogether. And so guess what?
That's right.
I've got Gardasil now too, like never having an abnormal pap smear again. This is why vaccines are great. And, we'll get into this a lot more, I think, tomorrow on our show. But I really, was encouraged by the fact that the advisory committee today, in advisory committee language, was actually recognizing that this is a big problem and that as colleagues, we can talk about evidence in our technical terminology. We can talk to each other using our secret expert language. And when we evaluate things, we should. But when we are communicating the decisions that we've made to the public and the reasons for doing it, we need to start using language and tactics that the public actually wants to listen to. And that was what was so striking, is that I like listening to the science, but even to me, a meeting like this would have been incredibly boring if I weren't like choking up at, the death of evidence-based policy development.
That's right.
It's really striking how boring it is when things are great. And I think the real challenge that we face is trying to figure out how to communicate how bad things would be without it, without sounding like, sort of the sky is falling, and putting people off, without using terms that have been appropriated by propagandists who are lying about the benefits that all of these things offer to people and to public health and science in general. I mean, we need to figure out a way to communicate to people in a way that gives them power and agency and, treats them as collaborators and partners, and not as just like, oh, this is the way we've been doing it and that's a status quo. Because I think we also run the risk of being criticized for having these sort of stuffy bureaucratic processes that are boring and inaccessible to the public. And then, you put something like this up next to Kennedy making a video of him like, drinking raw milk in a sauna with Kid Rock. And even though that's ridiculous, like, I even want to watch that more than a VRBPAC meeting, honestly. I mean, not really, but it's something that's going to grab a lot more people than a strong defense of evidence-based policy development and the scientific method.
Yeah, agree. This, fingers crossed, this is when we see this type of voting. But I'm grateful that it happened, and I didn't to watch all of the boring and juiciness. I'm grateful that we have to debrief, and I hope the folks will join us tomorrow. Taking that journey with you all. It seems like ACIP is still on track for next week as well, despite an agenda still not being published.
It sure is, and we are going to be promoting that more aggressively, which we always say that we're going to do, and then we don't because we both actually have other jobs. So we are going to do that though, and we are going to be covering it, and more about that to come.
All right. Well, always good to see you.
Always great to see you, and I can't wait to hang out tomorrow. So just one last thing before you go. You will see here that even though I'm not exactly very technically savvy with this, except I am totally into machine learning and AI and very sophisticated computational methods of analyzing virology data. But otherwise, I'm very bad at using things like StreamYard. However, you can see this platform has a lot of possibilities in terms of being able to share things.
Yes.
I can share slides and images and even little videos. And I've been experimenting with making some. So going forward, I hope to have some fun videos that might be kind of hopefully engaging for people to get pumped about ACIP, because ACIP is going to be the opposite of this. It is going to be insanity. And we are here for that. And so this was kind of a technical trial run and an opportunity for us to sing the glories of the VRBPAC as staffed by its current expert member roster. We will be here to talk about all the clowns on ACIP next week. And we'll see you tomorrow for taking shots. Thanks, Jeff.
Sounds good.
Great to see you.
Thanks, Angie.
Thanks,
See you
everybody, for
soon.
sticking with us.
Bye.
See ya. Oh boy, now I have to figure out how to end this.